Hey everyone, we're taking a look at the ear. When we say ear, I don't just mean the outer ear. The outer ear, the pinna or the auricle, which helps us get sound from a vertical plane and helps you locate things in where they are around you. Not just the outer ear, but also the middle ear, the middle ear and the inner ear. Middle ear is over here, inner ear is over there. So let's go over all these different parts. Once again, the outer ear, this is the auricle, A-U-R-I-C-L-E, auricle or pinna. It helps you locate sounds in the vertical plane. It also, protects the ear. You've got an opening right here. This opening, your ear canal, it's known as the external auditory meatus or external acoustic meatus or external auditory canal. Auditory canal is fine, but remember this is the external because the internal one is going to be over here. This is the internal acoustic meatus, internal auditory canal, where the nerve comes out. In the external acoustic meatus auditory canal, this is where you have earwax production. The term for earwax is cerumen. So these are ceruminous glands. If you recall, these are for these are a type of apocrine gland, specialized, kind of like the mammary glands are specialized apocrine glands. So as sound travels from the outer ear, it then will vibrate this thing. This is the tympanic membrane. You might know it as your eardrum. Tympanic membrane. The tympanic membrane vibrates, and then it vibrates your auditory ossicles. The first is the malleus, then the incus, which I can see better if you turn it. There's the incus, it looks like an anvil. And then this stirrup-shaped one, the stapes. This model shows it a lot better because it's much more magnified. Malleus, it's mallet-shaped. Incus, looks like an anvil. If you ever watch Looney Tunes, you know what an anvil is. Stapes, looks like a stirrup. It's the stapes that'll vibrate something on the cochlea. So this organ that's kind of snail-shaped. So this is what it looks like uncut. If you cut it in half, it looks like this. The stapes is vibrating what's called the oval window. So this is the oval window where the stapes would vibrate. And this snail-shaped thing, just from here and down, this is the cochlea. The cochlea, once again, is snail-shaped. So it's really just one long tube that curls over and over and over again. And so if you cut it, you'll see the chambers inside of it. It's a fluid-filled organ. You can see all these fluid-filled chambers here. From the outside, here's that snail shape, and then if we cut it along longitudinally, you can see the chambers here. There's distinct parts within this tube, and that tube curls around and around and around to the top. And that's why we see this repeating pattern of 3 small chambers. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. That repeats over and over and over again. It's colored here in pink, blue, and green for us. What are these different chambers, and how do you know which one's which? You can see that this chamber appears the biggest, and then there's this diagonal membrane that separates these two. Look for this diagonal membrane that's separating the compartments into 2. One's a bit smaller, one's a little bigger. This one here is scala vestibuli. Scala means duct. This is the vestibular duct, scala vestibuli. This middle one is scala media. This is scala tympani. So it just repeats over and over again. Vestibuli, media, tympani. Vestibuli, media, tympani. Over and over. This model is showing a cross-section of all of that. Scala vestibuli, media, tympani. Remember, this is just a cross-section of of this tube that's curling all the way around. So if we see in this longitudinal cut, we've got vestibuli, media, and tympani going around and around. So it's really complex, but it just circles round and round and round. I said that these things were filled with fluid. The scala vestibuli and tympani, they're filled with perilymph. Scala media is filled with endolymph. It's when the stapes vibrates the oval window and then causes vibrations, fluid waves in those fluids, particularly the endolymph of scala media. That's what would eventually leads to hearing sound. When you get fluid waves, pressure waves in the endolymph, we then get this thing to shift. This is called the tectorial membrane. And hopefully you've heard this term before, tectorial, like tectonic, like in earthquakes. Earthquake plates in the earth shift. Those are tectonic plates. So this thing shifts, the tectorial membrane. And when this shifts, it then moves and bends these little hair-like things. They're called stereocilia. These stereocilia are apical, specialized apical structures of hair cells. So it's labeled as 9 here. These are hair cells. They're called hair cells not because they produce hair, but they have hair-like things, stereocilia. The hair cells are the cells, the receptor cells that allow you to hear. They also allow you to sense your body position, but we'll get to that. Hair cells are neuroepithelium. So when these get activated, they then talk to sensory neurons. They themselves are not neurons. They are not nervous tissue. They are epithelial tissue. But they get activated when the tectorial membrane shifts over the stereocilia and bends them, activates the hair cells then activate sensory neurons. The cell bodies of sensory neurons are in this ganglion over here. And then these neurons continue on and form the cochlear branch of the vestibulocochlear nerve. Back to our model over here, you can see here's the cochlear branch of the vestibulocochlear nerve coming from our cochlea. That is how we hear things. One other structure that you should know, those fluid waves need to go somewhere, like the pressure needs to be relieved somehow. So after you get fluid waves going through scala vestibuli, they come out scala tympani, and that pressure gets relieved from a smaller hole. This is the oval window. Excuse me, what did I say? Round window. This is the round window, excuse me. Round window is where it escapes. Oval window is where we vibrate. Oval window, it's what the stapes vibrates. Round window is what allows that pressure to escape. The wave energy escapes out that way. All right. We have, besides hearing, the inner ear is really important for our vestibular sense. Our vestibular sense is our sense of body position. So it could detect rotational acceleration or linear acceleration. These semicircular canals, there are 3 of them, they detect rotational acceleration, which makes sense because they are round. At the base of each semicircular canal, you see this bulge here. That's called the ampulla. This bulge here is called the ampulla, and it's in the ampulla that you have hair cells, just like what we saw for the cochlea, except these hair cells aren't activated by pressure waves, they're activated by acceleration. So these semicircular canals are important for helping you detect rotational acceleration in the 3 major planes that you can turn your head. So that's for rotational acceleration, the semicircular canals. What we also have here are these 2 other organs, and inside of them, which we don't have pictured, but inside of them we have otoliths. Otoliths are these minerals that help you. They would basically, rather than having the tectorial membrane sitting on top of hair cells, you've got little rocks, tiny little rocks sitting on top of the hair cells. And those things will shift only if you linearly accelerate. Say that feeling you get when you're going quickly down or up in an elevator or on a roller coaster. That's the type of thing that you'd sense with these organs. These otolith organs, the one that's hanging down, that's the saccule for vertical acceleration. The one over here is the utricle for horizontal acceleration, like if you're stopping and going in a car. Altogether, utricle, saccule, and semicircular canals, they form the vestibular complex. So this is the vestibular complex. They branch to the vestibular cochlear nerve with their vestibular branch. So they come out here. They come out right here. That's the vestibular branch of the vestibular cochlear nerve. So this is cranial nerve 8, the cochlear branch and the vestibular branch. What else should you know? I think I forgot to mention some more features of the middle ear. So when we say outer ear, that's tympanic membrane and out. Middle ear is the border of the tympanic membrane and the auditory ossicles up to the round window. I keep messing it up, I'm so sorry. This is the oval window. Oval window. What else do we have in here? There is a muscle attached to the eardrum to help make sure that you don't damage your eardrum with loud sounds. Of course, there's limits to this. But we also have this passageway here. This is known as the auditory tube, also known as the eustachian tube. This helps you equalize pressure. This is air-filled. The external acoustic meatus and the auditory tube are air-filled. And so this connects to your pharynx, this connects to your throat. If you have an ear infection, this can often get filled with fluid or get blocked. So that's something to look out for. And it's also a reason why when you pop your ears, you can do that using your nose and your mouth. You're just increasing the pressure there to quote unquote pop your ears. All right, I believe those are all the important features I'd like you to know for the cochlea, for the vestibular complex, semicircular canals, saccule, utricle, malleus incus stapes, tympanic membrane, features of the outer ear, specific features of this cross section of a cochlea. Please let me know if you have any questions. Thanks for listening.